Hello everyone, this is Ketish here. In this video, I'm going to go through the AQA Combined Science Biology Foundation Tier, Paper 2. Question number 6 is on the topic of inheritance, variation, and evolution. Sexual reproduction in humans involves the production of egg cells and sperm cells. So we're talking about sexual reproduction here. Name the type of cell division that produces egg cells and sperm cells. Now, there are two types of cell division, mitosis and meiosis. The type of cell division that produces egg and sperm is meiosis. Meiosis is what produces eggs and sperm, which are totally different to each other. If it is mitosis, then it is basically, it's going to produce cells which are all identical, like in muscle cells. Sexual reproduction produces offspring that are genetically different from each other. Give two reasons why sexual reproduction causes variation in the offspring. Now, why would there be variation? Why would there be differences in the offspring? Offspring means babies, right? And variation is differences. So, give two reasons why sexual reproduction causes differences in the offspring. Well, that's mainly because the genes in the egg and sperm cells are not identical. Oops. Or you could say genetically different. That's why there is variation. And the genes, the second reason is the genes come from different parents. Meaning mother and father. They're not coming from the same parent. That's why they are different. Polydacty is an inherited disorder. Now, polydacty is basically where you get babies born with extra fingers. So you might have seen, generally we should have 5 fingers, well, 4 fingers and a thumb in each hand, right and left. Some babies might actually have an extra finger. They might be small, or sometimes it might be just fully 6 fingers. That's what polydacty means, right? Poly means many. The allele for polydacty is dominant. Dominant means it is actually strong, right? Right? Now, allele is means it's different types of genes. A person with 2 copies of the allele D will not have polydacty. Now, capital D is dominant. Lowercase d is recessive, meaning it's weak. A person with the genotype capital D is homozygous. Homo means same. That's why it's when it's identical alleles, we call it homozygous. What word describes the genotype, meaning the genetic makeup, which is this, capital D, lowercase d? Well, it's not identical, so we call it heterozygous. A person with the genotype capital D, lowercase d, and a person with the genotype lowercase d plan to have a child. Determine the probability that the child will have polydacty. Now, the probability that the child will have polydacty is if the child has capital D, or if they have capital D, lowercase d, then they'll still have polydacty. The only way that the child will not have polydacty is when they have capital D, lowercase d and lowercase d. So complete the Punnett square. Identify any offspring genotype that would have polydacty. So that's 5 marks. That's a lot of marks for this question. So let's, on the Punnett square, it doesn't matter where you actually put each of these. So I'm going to put capital D. What are the two? Capital D, lowercase d, and lowercase d, lowercase d. Capital D, lowercase d, and lowercase d, and lowercase d. So when you cross this capital D and this lowercase d, you get capital D, lowercase d, again capital D, lowercase d, and this d, lowercase d, and this lowercase d will give you lowercase d and lowercase d, lowercase d and lowercase d. So as you can see, 2 out of 4 is capital D, lowercase d, and 2 out of 4 is lowercase d and lowercase d. Now 2 out of 4 is 1 over 2, right, which is 0.5 or 50%. So you can write the probability in any way, but you would want to write it as 1 over 2 or 0.5 or 50%. The best way to write it is in the form of a percentage. So I would say it is 50% probability. You could also give it in a ratio as well, but the easiest to give it in the form of a percentage. Embryos can be screened for the alleles that can cause genetic disorders. So embryos are basically when a sperm and an egg fuse by fertilization in the woman's body, then the cell, fertilized egg cell, will develop into an embryo. This is the early stage of a baby. Now you can actually look at the cells from the embryo, right, and look at the genes. That's what means screen means, for the alleles that cause genetic disorders. Now we can actually look for certain types of genetic disorders. Now inherited disorders means that you already have, someone in the family already has it. So if you think someone in your family or you yourself have certain types of genetic disorders like polydacty, it's not the worst thing, but there are inherited disorders like cystic fibrosis that can be dangerous. So you'd want to screen the babies if you think you might actually have any of these inherited disorders that you don't really want the baby to have. Give 2 advantages of embryo screening. What is the advantage of embryo screening? Well, you could find out if the embryo has a genetic disorder, right? So by trying to find out whether the baby has a genetic disorder, that helps you to make some other decisions. What would these decisions be? Well, you could continue to have the pregnancy and have the baby, or you could actually terminate the pregnancy. So can help you decide if you want to continue with the pregnancy or have the baby, right? Now if you want to have the baby, then it can help you prepare for the baby with the disorder, meaning that you are going to be prepared when the baby is born. You're not going to be surprised, and you can actually make some life right choices in terms of finance and all of those things. That brings us to the end of question number 6, and I will see you in question number 7.